Hello, and welcome to our presentation on Adaptimmune.
Adaptimmune is a biotechnology company in the UK. Its goal is to develop affinity-enhanced T-cell receptors, which can be used in T-cell therapy for cancer. Adaptimmune uses a novel T-cell receptor method rather than the traditional chimeric antigen receptor method. Adaptimmune currently has a $350 million partnership with GlaxoSmithKline, one of the largest pharmaceutical companies in the world. Currently, Adaptimmune does not have any commercialized technology. Most of their products are in phase 2 of testing, going through clinical trials with human subjects.
So what is T-cell therapy? The process involves taking an individual's own white blood cells and infecting them with lentivirus. The lentivirus genetically modifies the white blood cells by injecting a new strand of DNA. As a result of modifying these T-cell receptors, these T-cells are more likely to respond to cancer cells. Adaptimmune takes a unique approach in T-cell therapy, but first we must understand the traditional chimeric antibody receptor method. This type of T-cell therapy focuses on selecting antigens that are favorable for attacking specific cancer cells. Adaptimmune's T-cell receptor method takes a step further. It uses a molecular selection system to optimize the affinity of the receptors, which increases the probability of attacking a cancer cell.
Adaptimmune's process of engineering anti-cancer white blood cells starts by taking a patient's white blood cells and then filtering that set of white blood cells for T cells. A bit upstream, Adaptimmune synthesizes antibody-covered beads in a mixer, and then mixes the antibody beads with T cells in order to enlarge the white blood cells and prime them for immunological activity. In a parallel branch of this process flow diagram, Adaptimmune passes white blood cell candidates, cell lines whose genetic code can allow for better anti-cancer T cells, through what amounts to a filter, in that the candidates who display enhanced affinity for a cancer antigen in question are sampled and then sent to the next process block. The next process block here illustrates what sets Adaptimmune apart from its contemporary counterparts. T cells that show promise are mutated at the genetic site that codes for their T cell receptors. The mutants are then filtered based on whether they are not harmed by the mutation and that they show even more affinity for a cancer's antigen than their non-mutant counterparts. From these supermutants, their DNA is then extracted and then fed into a reactor, which in this case is a heated cell medium with some cells in the medium, along with lentivirus to create a viral vector. The lentivirus infects the cells in the media, producing more viruses that take in the genes for enhanced antigen affinity so that we have a method of injecting our DNA into our patient's T cells. The viral vector is then mixed with a chemical called interleukin 2 to activate the T cell's immune response, along with the T cell along with the bead T cell mix, infecting the T cells with genes for higher affinity for a specific cancer antigen. The beads are then filtered from the mix, and the remaining mix is washed and then put in an evaporator, leaving behind the engineered cells with a bit of medium left. The cells are then refrigerated and stored until they are transferred back into the patient. The key unit operation, as implied before, was the mutagenesis of our cell candidates. Like the other unit operations in the Adaptimmune process, it is a batch process. The mutagen used here alters the genetic code for the T cell receptors, changing the final shape and thus the potential affinity for an antigen. This thus produces a handful of variants, which are then selected based on how improved their affinity for internal cancer cell markers are. This step illustrates perhaps the pros and cons of the Adaptimmune process in general. Because the unit operations are all batch, the process as a whole is slow, but easy to control and monitor to ensure the final product does not cause trouble in a patient. Due to the key unit operation in particular, this process at the same time also may have improved volumetric productivity compared to its competitors, since alternatives require more genetic engineering and thus more time to ensure a working final product. In the end, as the prevalence of cancer increases, the immunotherapy industry will most likely grow and become more competitive as companies begin to commercialize. Because patients' lives may be at the precipice when they undergo immunotherapy, volumetric productivity is key. The faster the cells can reach the patients, the more responsive patient treatment is, and thus the potential for profit.